And so our last talk of this session is by Ellen Hoffman, and she will discuss her work on estrogens suppress a behavioral phenotype in zebrafish mutants of the autism risk gene CNT-NAP2. So I think we can move forward with the green button.
Okay.
Yeah, and I think that's the pointer.
Okay.
Do you want to test it?
Okay, well, I want to thank the organizers for giving me the opportunity to present my work here today. And my research in my lab involves trying to understand the biological mechanisms underlying autism spectrum disorders. And autism is a group of neurodevelopmental disorders characterized by marked impairments in social interactions, communication, and the presence of restrictive, repetitive patterns of behavior. In particular, we're interested in how the growing list of autism risk genes shown here, when they're disrupted, disrupt the formation of specific neuronal cell types and neural circuits, leading to a complex clinical phenotype. And we're interested in leveraging this growing list of autism risk genes to identify novel pharmacological treatments that target core mechanisms in autism and are currently lacking. And so we use zebrafish as our experimental model system. We generate mutations in zebrafish versions of autism risk genes identified in humans using zinc finger nucleases, TALENs, and now CRISPRs. And we capitalize on two key advantages of zebrafish, which are well known to this audience. First, the ability to identify neuroanatomical phenotypes in brain development. And second, the ability to conduct high-throughput pharmacological screens to identify small molecule mediators of larval behavioral phenotypes. And our goal is to advance from risk gene identification to the discovery of novel pharmacological mechanisms. And I just want to note that our objective is not to recapitulate in the zebrafish the clinical features of autism, but rather to leverage the advantages of this system to advance from gene identification to the discovery of novel mechanisms. And what I'm going to talk to you about today is how we use this experimental approach with one particular autism risk gene called contactin-associated protein 2. Now, contactin AP2 encodes a neural cell adhesion molecule of the neurexin family, and we selected contactin AP2 to develop our model of the hundreds of autism risk genes because there's clear evidence from families from the old order Amish population, consanguineous families, that disruptive mutations in this gene cause a syndrome of autism and epilepsy. So we generated zebrafish mutants of contactin AP2. Now, zebrafish have two versions of contactin AP2. It's duplicated, contactin AP2A and 2B. And 2A shows a broader expression pattern compared to 2B, which is expressed more selectively in the telencephalon and diencephalon at 30 hours post-fertilization. And it's also expressed more selectively at 48 hours post-fertilization in the telencephalon, optic tectum, midbrain, hindbrain boundary, and hindbrain. So we used zinc finger nucleases to generate two early frameshift mutations in both paralogs. And we generated two lines of double homozygous mutants, and these mutants survived to adulthood, are viable and fertile. And both lines of double mutants we showed by Western blotting lose expression of full-length contactin AP2. So first, because an imbalance in excitation and inhibition has been implicated as a common feature underlying neurodevelopmental disorders such as autism, we were interested in studying the simultaneous development of inhibitory gabaergic and excitatory glutamatergic neurons in these fish. And what we found were that our contactin AP2 mutants, using this double transgenic line, show a deficit in the formation of gabaergic neurons. So what you're looking at here is a lateral and ventral view of dissected brains at 4 days post-fertilization. And they have a deficit in 4 brain GABAergic neurons. And we've quantified this and found that the mutants have about 34% fewer GABAergic neurons in this region compared to wild-type fish. In contrast, we didn't find any regional deficits in glutamatergic neurons. In addition, we found that the axon scaffold shown here in cyan forms generally normally. The only other morphological difference we found in the mutants was that they're on average about 18%, their brain size is about 18% smaller. Now, these findings were similar to findings in a contactin AP2 knockout mouse, which was studied by Dan Geschwin's lab at UCLA. And so we wondered how we could push the zebrafish model forward to identify novel pharmacological pathways. And so here we collaborated with Jason Ryle's lab at University College London, where he developed an assay for quantitative larval behavioral profiling. And in this assay, we in-cross our double heterozygous mutants, and then we pipette the larvae into the wells of a 96-well plate, and we track their activity, their locomotor activity on a rest-wake cycle in a light-dark cycle during days 4 to 7 post-fertilization. And then we quantify their behavioral output, their locomotor activity, and we genotype them afterwards. And what we found when we did this was that our contactin AP2 mutants show a selective phenotype of nighttime hyperactivity. And this was significant compared to background-matched wild-type fish, and we found that this worsens on successive nights. So we thought this was interesting because it's somewhat consistent with the loss of inhibition or inhibitory signaling. But then we asked, could we leverage this mutant behavioral phenotype to identify pharmacological pathways that may be disrupted due to loss of this autism risk gene? And here we used an assay called pharmaco behavioral profiling. This was developed by Jason Ryle during his postdoc in Alex Shear's lab. And what this assay does is it enables us to, what Jason did was he pipetted wild-type fish into the wells of a 96-well plate and then exposed them to a range of hundreds of psychoactive drugs. And then he was interested in studying zebrafish sleep, and he teased apart how these drugs affected different rest-wake cycle parameters that could be quantified. So for example, how active do the fish get during the day or during the night? When they rest, for how long do they rest, etc. And what he found was that he could use this assay to correctly classify drugs based on their pharmacological mechanism, based solely on their behavioral readout in terms of the patterns of how they affected these rest-wake cycle parameters. So as an example, you can see that alpha-2 adrenergic agonists cluster together, and they induce increased rest, and they decrease waking activity. And this is interesting because it's conserved compared to the effect of alpha-2 adrenergic agonists in humans, which are used to treat ADHD. So we reasoned that we could turn our contactin AP2 mutant behavioral profile into a behavioral fingerprint consisting of characterized by increased activity at night and decreased activity during the day. And then we compared this behavioral profile to a database of the behavioral profiles of wild-type fish exposed to 500 different drugs. And when we did this, we could predict drugs that could either exacerbate or suppress the mutant behavioral phenotype. What we found, not surprisingly, was that the contactin AP2 mutant fingerprint clusters with other drugs that induce nighttime hyperactivity. And we came up with a list of drugs that we called correlating drugs that phenocopy the mutant behavioral phenotype. Based on this prediction, we were able to design a mini pharmacological screen with two key objectives. First, we wanted to identify drugs that induce the most differential effects in mutants versus wild-type fish. And the goal of doing this was to identify pathways that are dysregulated in our mutants. Second, we wanted to identify, perhaps more importantly, we wanted to identify pharmacological suppressors of the mutant behavioral phenotype. So we selected 14 different drugs. 2 of them were strongly correlating with the mutant phenotype, and 9 of them were anti-correlating drugs, meaning that these drugs induce in wild-type fish the opposite profile to the mutant behavioral profile, and would be predicted to possibly rescue the mutant phenotype. Finally, we hand-selected 3 different drugs. Risperidone, due to its clinical use, it's FDA approved to treat irritability and aggression in autism, but it does not target the core symptoms. In addition, we tested zolpidem and diazepam, which are GABA-modifying drugs, because we identified a structural GABAergic deficit. So we exposed our mutant and wild-type fish to 3 different concentrations of each of these drugs in the larval behavioral assay that I described to you previously. And this led to 3 key findings. First, we found pharmacological evidence for dysregulation of glutamatergic signaling in contactin AP2 mutants. Specifically, we found that NMDA antagonists as a group were strongly enriched among drugs that correlate with the mutant phenotype. Similarly, we found that mutants display increased sensitivity to behavioral activation when exposed to increased concentrations of NMDA antagonists such as MK801. This suggests attenuation of glutamatergic signaling and was interesting because even though we did not observe structural evidence for regional glutamatergic deficits in our mutants, this was the first time that abnormalities in glutamatergic signaling were associated with loss of contactin AP2. Second, we found that of all of the drugs that we tested, GABAergic agonists induced the most differential responses in mutants versus wild-type fish. So what I'm showing here is a clustergram comparing the wild-type plus drug and mutant plus drug behavioral profiles. And what you can appreciate is that when we exposed fish to a GABA-A agonist, such as zolpidem, it induces very different effects whether you're looking at the mutant versus the wild type across our range of quantifiable rest-wake cycle parameters. And when we take out just one of these rest-wake cycle parameters, you can appreciate that a dose-response effect on the mutant is very different from its effect on the wild-type fish. And when we did a principal components analysis looking across all rest-wake cycle parameters, what we found was at the top of the list of drugs that induce the most different effects in wild-type versus mutant fish, we found that zolpidem and baclofen, which are both GABA-modifying, were at the top of the list. So this was consistent with our structural findings of abnormalities in GABAergic signaling in these mutants. And finally, and perhaps most interestingly, surprisingly, we found that estrogenic drugs were strongly enriched among drugs that anti-correlated with the mutant behavioral phenotype. So we found specifically that among the top 10 anti-correlating drugs, there were 4 drugs that had estrogenic activity. And this was interesting because contactin AP2 had not previously been associated with abnormalities in estrogenic signaling. One of these drugs at the top of the list was biochanin A, which was a plant-derived estrogen or phytoestrogen. And what was interesting was we found that low doses of biochanin A were able to selectively suppress the mutant behavioral phenotype. So what you're looking at in this clustergram is the mutant plus drug compared to the wild-type no drug profile. And what you can see is that low doses of biochanin A could actually bring the mutant behavioral phenotype closest to the wild-type no drug phenotype, in particular for rest-wake cycle parameters that define nighttime activity. And this drug rescued the phenotype with even greater selectivity than low doses of risperidone, which is FDA approved for treating aggression in autism. And when we did a principal components analysis looking across all rest-wake cycle parameters and all drug dose combinations, we found that the drugs that were able to most specifically suppress the mutant behavioral phenotype, we found low doses of biochanin A and low doses of beta-estradiol cypionate were able to selectively suppress this phenotype. And so using this unbiased approach, we were able to identify a novel, potentially novel pharmacological pathway associated with an autism risk gene for further investigation. So in conclusion, with respect to our contactin AP2 mutants, we found that they have structural deficits in GABAergic neurons, a selective behavioral phenotype of nighttime hyperactivity. We found pharmacological evidence for dysregulation of GABAergic and glutamatergic signaling. And most interestingly, we found that estrogenic drugs are able to selectively suppress the mutant behavioral phenotype. And now we're very interested in the mechanism by which the estrogens rescue the phenotype. And we're currently doing functional imaging to look at this in more detail. We have some preliminary evidence that the estrogenic drugs like biochanin A likely act acutely, and we hypothesize that they may be acting upstream of the gabaergic and glutamatergic deficits. And in conclusion, in my lab, what we're doing now is we've now generated zebrafish mutants in all of the autism risk genes that you can see on this slide. And I hope that some of you had the chance to stop by my students' poster. Sindis Ijaz presented a poster looking at some of the phenotyping that we're doing on these other lines. And what we're doing is we're feeding them forward into the same pipeline that I showed you for contactin AP2, looking at structural abnormalities, and also looking for the identification of convergent pharmacological pathways with relevance to autism. And I just want to thank the members of my lab and my mentors turned collaborators, Matt State and Antonio Heraldes. I developed this work in their labs at Yale. My collaborators, Jason Ryle and Steve Wilson at UCL, and Mikhail Granado, Herwig Baer, and Michael Baresi. And if students found this interesting, please see me after the talk. And I just want to mention briefly that Sadie Bergeron and I started a zebrafish neural disorders research interest group on LinkedIn. So if this is of interest to you, please find us on LinkedIn. And thank you.
That was really beautiful. With the caveat that that zebrafish mutants and autism genes aren't necessarily going to exactly phenocopy the human disorders, have you looked for any sensory integration defects in the larvae, responses to startle or habituation, that kind of thing?
Yeah, so that's a great question. So for the contactin AP2 mutants, we did do additional behavioral tests. We did look at startle and habituation. For the most part, their SLC and habituation and pre-pulse inhibition was normal, but they did have a delay in their LLC, or the sort of delayed startle response. It was a subtle phenotype, and we didn't investigate that further. But otherwise, in general, their startle responses were generally normal or unremarkable. But we are interested in looking at that in some of our other mutants, where that may be even more relevant.
Thank you very much. So that's the end of our disease models session, and I would appreciate it if you could help me thank the speakers for the really great session. So thank you.